Hi, teacher. We are Ronald Manosalvas and me, Alex Valdivieso, and today we are speaking about the calculation of G values in e-coli using UV-Vis spectroscopy. Well, what is the UV-Vis spectroscopy? Uh, The UV-Vis spectroscopy refers to the measurement of the absorbance of ultraviolet visible light by E. coli bacteria to determine its growth and viability in experiment. G values are a measure of bacterial growth rate and are calculated using absorbance measures and different wavelengths. This method is useful for monitoring bacterial growth in real time and evaluating the effects of various treatments or environmental conditions on the bacteria. Objectives. For this practice, the objectives were learn to calculate the generation time of the microorganisms from growth data, understand the importance of the generation time in the evaluation of food quality and safety, analyze how different environmental conditions affect the generation time of microorganisms in foods, and the specific objectives were become familiar with the materials and equipment necessary to measure microbial growth and calculate generation time. Prepare microbial cultures in optimal growth conditions. Measure microbial growth at regular intervals. Graph microbial growth data as a function of time. And finally, calculate the generation time using the appropriate equation. Materials and methods. uh, Inputs. For this practice, we needed microbial strain of interest, suitable culture medium, test tubes or flasks, Erlenmeyer, weather bath or orbital incubator, spectrophotometer, pipettes and tips, and a pipetter.
Okay, now explain the methodology for this experiment. Okay, the first part is the preparation of the bacterial culture. They grow a strain of E. coli in suitable culture medium until reaching the exponential growth phase. It is important that the culture is well aerated and in optimal condition for cell growth. Detection of the cell's density measures the optical absorbance of the culture sample at a specific caliber using a UV visible spectrophotometer. The commonly used wavelength for E. coli is 600 nanometers, but the choice will vary depending of the experiment these conditions. Be sure the celerity in the spectrophotometer they correctly before measurements. The preparation of the calibration curve. Prepare a series of dilution of the culture samples, obtain a series of the absorbance values that cover a linear range of the concentrations. Measure the absorbance of the dilutions and record the values. The calibration curve constructions plot absorbance value on the y-axis against no cellular concentration on the x-axis. Fit a line best of the experimental points to obtain a calibration curve. Finally, calculating the G value. Once you have this calibration curve, you can use the calculate the G value for E. coli samples with you have the measured absorbance. Simply plug the absorbance value in the calibration curve equation and so obtain corresponding cells conservation. Okay, now explain the results of this experiment. In the first part of this experiment, it's failed because of different factors that affect for these results. Observe that in the absorbance, uh, 50 to 500 observes the different points in different parts that this graphic doesn't represent of the exponential curve. And in the same time, pass with the absorbance to 16 points. The next week, uh, obtain the next results to this experiment and compare these results with another part. And it's possible that affect different factors for temperature, pH, time of the take the time for lecture. Absorption is very different to another experiment, and it's possible that affect the similar conditions or the making of the experiment. Okay, now explain the annexes. Okay, the first question is why is important to know the generation time of microbiologicals in food microbiology? Okay, to explain this part, the spectroscopy experiments, especially when working with microorganisms of cells and culture, generation time is crucial parameter that is related of the exponential growth phase of the growth curve. The exponential growth phase is on the wisdom microbiologics or cells are actively multiplied at a constant rate. During this phase, the population doubles in a specific time, which is the generation time. In other words, the generation time is time required for a population double. Okay, the next question is how do your environmental conditions, temperature, pH, nutrients, concentrations influence the generation time of microbiology in food affect? Okay, the, these problems affect the medium because they, for example, if some bacteria stay in different conditions, they disinvolve the different forms. It's possible that some bacteria die and some bacteria in other conditions affect and float in the medium. Okay. It's possible that if the control of these bacteria in food is very interesting in the control because some people eat today all of the different products of the uh to life. Okay. What is the relation exists between generation time and the exponential growth phase in the growth curve? Okay. The measured absorbance of 50 550 nanometers is technique used to estimate the cell density in microbiological cultures. Here are some important reasons to take these measures. The quick and easy. Absorbance mixture is quick and easy method to estimate the cells density in a microbiological culture compared to other laboratories methods, such as cell counting by microscopy and non-destructive. The absorbance measurement is non-destructive, meaning that the sample is not required to be certificated to perform the measurement. This allows continuous monitoring of the cell's growth, never in the same temperature. Okay. The correlation with the cell's density. There is a linear relationship between the measured absorbance and cell density in many types of microbiological cultures. This means that absorbance reading can easily be converted into estimate of cell density, proving an quantitative way to assess cell growth. In summary, in measuring absorbance of 550 nanometers is valuable use technique for estimating cell density in microbiological cultures due to rapidity, easily low cost, and ability to provide quantified measures of the cell's growth in the real time. Next, please.
What relationship exists between generation time and exponential growth fast in the growth curve? The generation time directly influences the duration and intensity of the exponential growth phase in a growth curve. A shorter generation time corresponds to a faster growth rate and a shorter exponential phase, while a longer generation time leads to a slower growth and a longer exponential phase. Next. What other methods can be used to measure microbial growth and calculate generation time? We have 7 methods. We can use the plate count method. This involves spreading a sample of a microbial culture onto an agar plate and counting the number of colonies that develop after incubation by tracking colony growth over time. One can estimate generation time. 2. Turbidity measurement. Turbidity or cloudiness of a liquid culture is measured using a spectrophotometer. As microbial cells grow and multiply, they increase the turbidity of the culture. The time it takes for turbidity to double time can be used to estimate the generation time. 3. Biomass measurement. Biomass can be measured by weighing the cells or by quantifying cellular components like proteins or DNA. By tracking biomass increase over time, one can calculate generation time. 4. Microscopic cell counting. Direct counting of microbial cells using a microscope and a counting chamber or hemocytometer by counting cells at regular intervals. One can estimate generation time. 5. Flow cytometry. This method involves passing a microbial sample through a flow cytometer which can count and measure properties of individual cells in a population. It allows for high throughput and accurate measurement of microbial growth dynamics. 6. Real-time PCR, polymerase chain reaction. PCR can be used to quantify the amount of DNA in a sample at different time points, providing insights into microbial growth dynamics. 7. Bioluminescence assays. Some microbial species express bioluminescent proteins that emit light in the presence of specific substrates. By measuring light emission over time, one can monitor microbial growth. That's all. Thanks.
Thank you,
Uh,
teacher.
bye.